Hello everyone, welcome to the class of biotechnology. My name is Dr. Farah Maarfi and I hope you all are doing well. In our last lecture, we were talking about differential preparative and ultracentrifugation that separates the subcellular fraction on the basis of size, shape and density, right? Size, shape and density. And another parameter that was important there was constantly increasing speed, that is from 1000 G to 1 lakh G, right? So here as shown in figure, when increasing centrifugal force is applied on the homogeneate, then the particle according to the size, shape and density will separate out, right? When a centrifugal field is applied to this tube, then the particle according to their size, shape and density will get settled down at the bottom of the tube. As you are seeing here, the larger one will settle out the bottom of the tube first under the influence of the centrifugal field, followed by the medium and the smaller one. Smaller, medium and the larger particles, right? So the applied centrifugal field size, or you can say mass of the particles, are inversely proportional to each other. They are inversely proportional. Means larger the size, lesser would be the centrifugal force, then greater would be the sedimentation rate. Greater would be the sedimentation rate in comparison to the smaller ones. Smaller particles required greater centrifugal force. Smaller particles required greater centrifugal force, right? In order to settle down at the bottom of the tube. So these particles required less centrifugal force in comparison to these particles so that it gets settled down at the bottom of the tube. So up to this, the size was the most important factor, or you can say the most important parameter that decides the rate of separation. But there occurs a situation where the particles are almost similar in size. Similar in size. And hence you cannot separate these particles on the basis of their size only. So here comes the role of density. The exploitation of the density is required here. For example, the mitochondria and lysosomes, they both are of same size, or you can say they both are of almost same size. Its size is of around 0.5 to 10 micrometer, right? But they differ in their density. Mitochondria is denser than lysozymes. So density of the mitochondria is more in comparison to the lysosomes. It is more denser, less denser, due to the presence of proteins or lipid molecules. And here mostly lumen and some digestive enzymes are present. Another example is ribosomes and ribosomes. Ribosomes. They both are of almost size 100 to 200 nanometer. There are many other examples also. These are just a few of them. So here comes the Role of density gradient preparative ultracentrifugation. They works on two methodology, that is rate separation and equilibrium method. Equilibrium method. Let's see how these two methods are being employed. So as you all know that the density gradient centrifugation let's see how these two methods are being employed here. So as the name suggests, density gradient centrifugation, that means we are creating a gradient here, right? We are creating a gradient here. Gradient of what? Gradient of density. Gradient of density. So density gradient can be generated by placing layer after layer of the gradient medium. It can be generated by placing layer after layer of the gradient medium in any test tube. That gradient medium can be sucrose or cesium chloride or sodium bromide, sodium iodide, etc. Ready to use gradient medium is also available in the market. Cesium chloride is used for the binding of the DNA and the isolation of the plasmid, nucleoproteins and the viruses. Sodium bromide is used for the fractionation of the lipoproteins. Sodium iodide for the DNA and RNA molecules. These are the gradient mediums that you can prepare by yourself in any lab. Several few ready to use gradient mediums are also available, that are percol, ficol, dextrane, metrizamide and nicodens. Let's understand this, that what actually we are doing in density gradient preparative ultracentrifugation. Suppose this is a tube containing a layer of density gradient medium. Layer of density gradient, right? And we place a sample at the surface of this layer. Sample at the surface of this layer. And after placing the sample at the surface of this layer, we apply a centrifugal force to it. Here, the applied centrifugal force will be same throughout the process. That is around 1 lakh G. So before moving forward, we should keep two things in mind. The first one is that the density of the medium is not same. You can say at the top of the medium, the density is less. And when you move towards the bottom of the tube, the density gets increased. Second thing is that we should choose such gradient medium that has density lesser than that of the sample. That means the sample should have density greater than that of the density of the medium. So density of the sample should be greater than that of the density of the medium. Suppose this is the order of the density of these particles. The gray one is the more dense and the orange one is the medium dense and the green one is the lesser dense. Suppose this is the order of the density for these particles. The gray one is the more dense one and the green one is the lesser dense particle. So when you apply the centrifugal force to it, for a definite period of time, then the gray one will sediment out first at the bottom of the tube, or where the density of the medium get equal to the density of the particle. Here the rate of the sedimentation will be 0. this will occur only for a particular specific period of time. So time here is the very important factor, because there is more other particles also present in the tube at the same time, but they resides at the uppermost layer of the tube. They did not goes to the they did not go to the bottom of the tube, because the density of the particle is not equal to the density of the medium here, right? But for the gray one, for a specific period of time, the density of the particle get equal to the density of the medium here. That's why it gets deposited at the bottom of the tube. But if you increase the time further, then all the particles, that is the green one and the orange one, will also get deposited out forcefully. This you can say that is the major drawback of the density gradient centrifuge also. So this type of centrifugation is called the rate zonal centrifugation. Centrifugation. It is a type of density gradient preparative ultracentrifugation, because here the centrifugation speed is 1 lakh G, or you can say the applied centrifugal force is 1 lakh G. But it depends on the time, and if due to any reason time increases, you cannot obtain the required interested particle. So to avoid this limitation, we perform isopycnic centrifugation. Isopycnic density gradient preparative ultracentrifugation. Again, the speed will be 1 lakh G. That is, the applied field with applied centrifugal force will be 1 lakh G. Right? But here only the important factor or important parameter is density. Density of the particle is the only factor that plays a role here. Right? Let's understand this how. Suppose this is a tube and these are the samples of different density but of same size. Okay? Here the density is different for these samples. Different density. And here at the bottom, the gradient mediums are divided into different layers. These are the different layers of the gradient. Suppose here the density of the medium is 1.4, here the density of the medium is 1.2, and here the density of the medium is 1. So at the bottom of the tube the density is greater, and at the top of the tube the density is lesser, right? So when you apply the centrifugal force to this setup, then those particles that have density around 1.4 will go to this particular layer of the medium, right? Like this. Here suppose the gray one is having a density 1.4. So here the density for the green will be equal to the the density for the gray will be equal to the density of the medium present here in this layer, right? After that, this layer of having 1.2 density, then here the orange one, the density of the orange will get equal to the density of the medium, that is 1.2. So that means ki the density of the orange particle is around 1.2. After that, this one, the green one, the density of the green will be equal to the density of medium, that is equal to the 1. So in this fashion the particles deposited out, and you all know that as soon as the density of the particles get equal to the density of the medium, then the rate of sedimentation will be equal to the 0, and here the sedimentation stops, right? Another example, suppose you want to separate the DNA from a mixture of samples. right? And you know that for the separation of the DNA, the medium of choice is cesium chloride. Using the gradient of the cesium chloride, you can separate out the DNA from any mixture, right? So suppose this is a tube. In this tube, you will generate or you will create the layers of gradient of what? Cesium chloride. Cesium chloride. So at the bottom of the tube, suppose you have created a density of around 1.75, 1.70, 1.65 and 1.60, right? And then you place the sample at the top surface layer of the medium, right? After applying the centrifugal force to it, the DNA that have a density, you know that the density of the DNA is around 1.70. So after applying the centrifugal force, the DNA will goes to this particular layer, right? The DNA will goes to this particular layer. Here, the density for the DNA will be equal to the density of the medium, that is around 1.70, and hence the sedimentation rate will be 0 here. So in this manner, you will able to separate the DNA out of mixture. So this was all about the today's lecture. This is the flow chart for the type of the centrifuge from the starting differential centrifugation to the isopycnic centrifugation. If you like this video, please like and subscribe. In the next session, we will study about the analytical centrifuge. So till then, thank you so much.